Good morning to all the speakers, delegates, students, and participants present here today. Professor Kobayashi and me welcome you all to the first live session of the second International Electronics Conference on Applied Science, organized by Applied Science Journal of MDPI. I am Nunzio from University of Campania, Luigi Vanvitelli in Naples, Italy. I would like to thank the scientific committee, the invited speakers, the reviewers of the submitted works, the session shares, the MDPI staffs, and all the authors of these works. In particular, we have received about 100 works that after this conference could be submitted in extended version for a publication in the special issue of the journal Applied Science, entitled Selected Papers from the Second International Electronics Conference on Applied Science. I thank you all to be here today. We would like to thank MDPI Group for having supported this interdisciplinary summit on the applied science. In these days, very interesting talks will be presented by experts on different topics, and all the accepted work can be read in open access mode on the website of the conference. Now we will begin the first live session. I would like to invite the session chair, Professor Rogerio Noguiera from University of Aveiro, Portugal, to start this live session. Thank you.
Okay, thank you, Nunzio, and also Charlotte. So thank you everyone and all the speakers and all the audience that is connected here. It is a pleasure to share this session. Now we are going to start for the first speaker, which will be Cosimo Trono from National Research Council, CNR-IFAC from Italy, with the presentation. It's called Biosensing Systems Based on Unconventional SPR on Fibers, the Multimode Plastic Fiber and the Light Diffusing Fiber Examples. So the floor is yours, Cosimo.
Okay, thank you, Rogerio, for your presentation. I will describe in my talk the activity developed in collaboration with the University of Campania, the Department of Engineering. I will talk about two examples of surface plasmon resonance sensing developed on optical fibers. A very brief introduction about the SPR. SPR is a surface plasmon resonance, is the interaction of an electromagnetic wave with the conduction electrons. This is the classical Kretschmann configuration in which we have a dielectrics and transparent dielectric material on which is deposited a layer of a metal layer, and under right condition of the wavelength and incidence angle, we can have the transfer of energy from the electromagnetic wave to surface plasmon modes of conduction electrons. On the right side, there is some example of the implementation of these techniques on optical fibers. In this case, we have to expose the core of the fiber and to deposit the gold, the metal layer on this exposed part in order to have the interaction of the guided light with the metal layer. So there are several possibilities. For example, to expose the core by etching or tapering the fiber, or to deposit the metal structure on the face of the fiber in correspondence of the core, or by side polishing the fiber. So the first example is constituted by a plastic optical fiber. This is a 1 millimeter fiber, very, very cheap. A part of the fiber is embedded on a resin block that can be polished. In this way, we have the exposing of the core of the fiber. We have a flat surface on which it is possible to deposit a photoresist buffer and to deposit on the photoresist a gold film. Here you can see an image of the sensor. This is the fiber and this is the cylindrical resin block, and you can see here a pictorial schematization of the sensor. Second step is the realization of a system in order to have an optimal mechanical, thermal, and fluidic stability. This is a macrofluidic flow cell constituted by a metal aluminum bottom part with an housing in which the optical fiber sensor can fit, and a top part made of PDMS in which is realized an inlet and outlet and a microfluidic channel exactly in correspondence of the exposed part of the optical fiber. This is the experimental setup. Again, very simple. We have a white light source, which an halogen lamp, and the light is injected in the fiber and is guided by the fiber, and the exposed part. In the exposed part, there is the interaction with the metal layer, and the signal is acquired by means of a spectrum analyzer in the visible range. Here you can see an image of the setup with a thermoelectric cooling system in order to stabilize the temperature in the system. We have realized a bioassay experiment on the fiber. On the left, there is the description of the immobilization phase with a functionalization of the gold layer surface and a covalent immobilization of, in this case, anti-CRP capture antibody, followed by a passivation of the surface with the bovine serum albumin. On the right side, you can see the behavior of the resonance of magnified part of the resonance, and the calibration curve realized in human serum matrix. The second example is a non-conventional fiber. Usually the fiber, the aim of the fiber is to guide the light at the best. In this case, Corning has realized this fiber with the scatterer inside the core, and so the light is diffused from the side of the fiber. And so the idea of our colleagues from University Vanvitelli, the idea of Nunzio Cennamo, was to use this behavior of the fiber by realizing an SPR sensor directly on the surface of the fiber. So in order to use this capability of the fiber. The sensor is realized by removing part of the polymeric cladding of the fiber and by deposit a gold layer on this part. The length of the sensitive part is 20 millimeters. And again, we have realized a system for stabilizing the temperature and the mechanical part, and to have an optimized fluid interaction with the sensing part. In this case, we have two parts, a metal bottom part and PMMA top part, and the fiber is housed in between these two parts. And when everything is sealed, we have in the PMMA a microfluidic channel with an inlet and outlet. So we have a closed fluidic system. And again, it's possible to control and monitor the temperature of the system. The experimental setup is again the same, a white light source and a spectrum analyzer in the visible range. This on the right now, there is a typical resonance obtained with this system. The red one is the resonance normalized between 0 and 1. So now the issue is to extract a number, the wavelength in correspondence of the minimum of the resonance. Usually the simplest way to describe the resonance in the Kretschmann configuration is by means of a symmetrical Lorentz function. In a work, Noguiera and his colleagues derived a more realistic description by modeling the interaction with the three-layer Fresnel equation, in which the three layers is constituted by the glass prism and the metal layer and the sensing layer. So to have a brief look of this equation, there are 6 parameters, but it's interesting to have a look at the ratio between the second and third, B and C. In particular, if B is greater than C, more B is greater than C, then more D curve is similar to a Lorentz function. And in case, in the other case, we have a not symmetric curve. So our hypothesis now is that we have a very different system with respect to the Kretschmann configuration, in which everything is controlled, the angle, the wavelength, and very, very precise system. Here we have a fiber with a lot of scatter, and so a very chaotic behavior of rays inside the fiber. Rays are scattered and sometimes are reflected by the gold layer, can interact with plasmon, and at the end exit out from the fiber. So the hypothesis is that the transmission is anyway proportional to the refraction on the gold layer, so proportional to the Noguiera equation. So the hypothesis is that what we measure from the output of the fiber, it can be in some way described by this kind of equation, which is practically the same. Here, simply the parameters are arranged in different way in order to speed up the fitting procedure. And here is our resonance, and the black curve is the experimental curve. The blue one is the fitting by means of this equation, and we can say surprisingly the fitting is good, surprisingly, considering that we have a very, very different system with respect to the Kretschmann configuration. We have a chaotic system, random, and in comparison with a very deterministic system constituted by the Kretschmann setup. Anyway, we want to have more precision in the calculation of the minimum wavelength, and so we restrict our analysis to only the bottom part of the curve. Here you can see the magnification of the spectrum, and the blue curve is this one, and the red one is the fitting only on this part of the spectrum. So now we can extract the minimum wavelength by calculating the minimum of this equation, in which the parameters are obtained by means of the best fitting procedure. So again, we performed two bioassays on this fiber, an EGG, anti-EGG bioassay, and a CRP anti-CRP bioassay. This is the immobilization sequence, the functionalization of the fiber in order to have carboxylic groups on the gold layer, and the capture antibody immobilization on the surface, and the passivation of the gold surface. On the right, you can see the shift at the end of after the end of the capture antibody immobilization. You can see that the resonance are different because we are using two different sensors in this case, and the shape and the position of the resonance is strongly influenced by the gold layer and the deposition of everything, and so we don't have exactly the same spectral characteristic for every realized sensor. This is the EGG, anti-EGG bioassay. This one was realized in PBS. On the right is the magnification of the first 5 points. And the red dot represent the test of a non-specific interaction. We flow, in this case, anti-CRP antibody, and we can see that we don't have a grid shift, so the biochemical layer work fine from the specificity point of view. And here we have the calibration curve in case of the CRP. In this case, we used a more realistic matrix. The CRP was spiked in CRP-free human serum, which is a very complex matrix with respect to the PBS buffer. The performance are very good, in particular in terms of limits of detection. In this case, the comparison of the plastic optical fiber sensor that I described before, we have a better performance in terms of limits of detection. So, in conclusion, I described 2 examples of simple and non-conventional system obtained by using very cheap fiber and very non-conventional fiber. The first one, a plastic optical fiber, and the second one, a light diffusing fiber. In both cases, we realized systems for the stabilization of the external perturbing elements. And in the second case, we realized also a technique for the extraction of the minimum of the resonance wavelength. And both cases we performed bioassays to demonstrate the capability of the 2 systems in this sense. Okay, this is the end of my presentation. Thank you to everybody for hearing me.
Thanks a lot, Dr. Cosimo Trono. Trono. It was a very nice indeed presentation. For the first time, we have this real demonstration of a sensor with these diffusing fibers, which is very interesting. We have here one question from the question is, how advantage are the LDF over conventional POF? Do you want to comment?
Okay, yes. In this case, I mean, the idea was that the aim of the LDF fiber is to couple a lot of light out from the fiber, from the lateral side of the fiber. So the idea is that in this way there is also an increment of the number of optical rays that can interact with the metal layer with respect to the conventional case. And so this was the point to use this strange behavior of the optical fiber, different behavior from the classical one, which is to the guiding properties, the best guiding properties of the fiber.
Okay, thanks a lot once again, Dr. Cosimo Trono. Now we thank you. Now we move to our next invited speaker, which is Dr. Raghavendra Sai from Indian Institute of Technology in Madras, Chennai in India. And the title of the presentation will be Plasmonic Fiber Optical Absorbance Biosensor, a point-of-care platform toward atomol and light detection.
Good morning. Thank you for giving me this opportunity to share our work with some of the peers of this field. I know I actually have gone through all your papers, and it's a great pleasure to come here. This is India, and we are in Chennai here in southern part of India. Our campus is also known for nice wildlife. Of course, not really wild, but here. So we are very much aware of this lateral flow assays glucose monitoring kits. They have changed our life to some extent, but we still have this blood. We go to clinical diagnostic labs to give samples and get the results. So we actually want to see if we can bring some technologies to actually onto the left side, so that we have this diagnosis done easily, just like glucose and pregnancy. So in this we are doing it mainly to keeping India in mind. And what we do today in this lab, biosensors lab, is basically to develop sensing technologies for clinical diagnosis, water, environment, food, pharma, and agriculture. So our aim is that where we want to hit these applications, we make use of optical fibers and plasmonic particles and develop devices. So current point of care devices, undoubtedly lateral flow assays. We are very much aware of how they work, and also aware of the limitations of the low sensitivity and dynamic range. So keeping those in mind, we thought, whether can we use optical fibers as transducers to develop sensitive biosensors. Here, we are very much aware of this events and wave phenomena, and then from Francis Ligler's paper, it's a very well-known demonstration of how fluorophores can be excited using events in field to actually realize a fluorescence-based biosensor. So what we are looking at is to see if we can realize a fiber optic absorbance biosensor rather than a fluorescence-based biosensor. For that, if we take a decladded fiber, the sensitivity, the events and wave is pretty, small. So by modifying geometry, of course, D-shape, tapering, all these help. But what we have taken is, we have actually taken a U-bent optical fiber. So this study, particularly in this particular figure, image shows a silica optical fiber, 200 micron fiber, bent to have a bend diameter of 1.5 mm. So once you functionalize it and dip it in fact, one advantage of this U-bent probes is they are so small that you will be able to dip them into an Eppendorf tube or microcentrifuge tube. So as you do that, when you put amine-coated amine functionalized probes into gold nanoparticles, the optical absorption spectrum of gold nanoparticles is faithfully reproduced. As more and more nanoparticles come and bind, you actually see huge absorption over time. So what's really happening here is, in fact, as we bend the fiber, basically what's happening is a part of the light is actually lost due to refraction, but a significant amount of up to 60% of light is still there in the fiber, and it continues to propagate further, right? So that means basically, and not only that, most of this light is actually concentrated around the outer curvature, and that's how when we have something stuck to the nanoparticle stuck to the fiber, we see a great absorption response. Try to realize a sandwich immunoassay with gold nanoparticles, just like the way it happens in a lateral flow assay. We try to do that on a fiber surface. Here, what we took is a very simple setup. We have a custom light source, and it's a Thorlab photodetector, and these are bare fiber adapters, and then basically we have this U-bend probe. So, and then subsequently we subjected it to different conditions. In fact, the incubation step is for 5 minutes, followed by dipping the probe into this tube while observing intensity in real time, intensity of the photodetector. So to our surprise, we were able to go down to human IgG concentrations as low as 1 femtogram per ml. This is the control line, gray line, and 1 femtogram per ml is actually above the control, detectable, but not really, may not be quantifiable. So to further confirm, what we have done is we actually, once the nanoparticles have formed on the fiber surface, we actually did the silver enhancement, and then what does this silver enhancement do is basically it will silver gets deposited around the nanoparticle. It actually leads to increase in absorption. So in the previous slide, you see the time from 0 to 21 minutes. At the end of 21 minutes, basically these probes are subjected to the silver wash and silver solution, silver enhancer. So we actually see again a proportionally increase in response, and that confirms that this assay is working. And you know that when we actually have such concentrations, we actually have molecules as low as about 150 molecules in 25 microliters. So not only that, here in this study, we have used 40 nanometers. In fact, one can actually use 13 nanometers to probably shift the dynamic range as well for the suitable analytes, for the required analytes. So we just compared this response with what Chad Merkin has done in 2003, the first attomolar analyte detection paper. So in 2 steps, we are able to achieve this thanks to the gold nanoparticles, extinction coefficient very high, and the very high absorbance sensitivity of these U-bend probes. So we do get close. We are able to go down to 6 attomolar, not 3, but of course, this is a phenomenal paper, and we are able to go very close to it. The only thing is only in 2 steps instead of 4 or 5 steps. So we just confirmed this with SPR. So the volumes involved are only 50 microliters. So what you see is only 50 microliters here. So the response, rise in response because of 50 microliters is what you see. And then we see that what we are getting is, we confirmed it, that we are getting an appropriate result. So we just looked at assured criteria, affordable, all that WHO criteria. We seem to, we would like to go in that direction. So we will see how it goes. In fact, I just want to bring it to your attention that our review manuscript on attomolar analyte sensing technologies just got accepted, and the proof is just submitted. Hopefully you will find it interesting to read. It's accepted in Chemical Society reviews, so I hope you'll find it useful. So we have applied the same assay. We call it as PFAB, plasmonic fiber optic absorbance biosensor. So this one PFAB to TB screening using MANLAM as an analyte, and we got a similar result that again establishes that this platform does work in automolar and analyte range. So we have been working on plastic optical fibers as well. Here you see that one of our paper where we actually showed that this bending, the bend diameter of the fiber and for a given core diameter is the optimum to get a high refractive index sensitivity. This has been very well received thanks to the scientific community. So we have been doing some studies where we want to develop SPR and LSPR probes that also has been published in JRS. So one thing, one interesting thing that we find is when we actually try to put these plastic optical fibers in a vacuum chamber, basically the water pops out, water bubbles basically pop out, and they leave such kind of nice structures on the surface. So maybe I don't know whether it's a hindrance or aiding to the sensitivity. So we have also done, we have to publish it. One thing is basically allow us to monitor what is, and their shape transformation we'll be able to observe in real time. So this is one of the very important advantages of these probes. So coming to technology development, we have been making these probes manually using candle. Then we moved on. We started using a laser to make these probes. We have automated this process. We are able to make it now very well. So this is a POC device. You know, we actually want to bring this entire to a palm-based device, palm top sensor. So we kind of going ahead. There are some roadmap. We want to see how we will be able to apply this technology for clinical diagnosis, water, and food monitoring. So thanks to the team and thanks to the funding agencies, collaborators. Thank you very much.
Thanks a lot, Dr. Raghavendra Sai. Very nice presentation. Let me check if we have any questions. Don't have, but actually I have one question. Is regarding when you use the POF for the optical sensing, it absorbs the liquids. So how do you handle that those sensors to monitor in different ways, or you just use them once, or do you have to do any kind of procedure to use it again?
POF, yeah, we have to use it only once, discardable. Yeah.
Okay.
Yeah, liquids, so we cannot use it more than once. Yeah, and also we have to compensate for the these.
Sorry, we are having difficulties with your microphone. Could you?
We have to compensate for the changes caused by the liquid absorption by the probe. So these things we have not yet worked on.
Okay. Okay. Thank you.
Thank you.
Thanks once again. Now we are moving to our next invited speaker, which is Laura Pasquardini from Indivenire in Italy, and the title of the presentation will be Aptamer-based optical biosensors, a focus on plasmonic plastic optical platform. Dr. Pasquardini, please, the floor is yours.
Okay, thank you very much for the introduction and also for inviting me to this conference. The topic of my talk is to show you the coupling between aptamers and POF-based devices. This work was made in collaboration with Dr. Cennamo, Luigi from University of Vanvitelli in Campania. And just a few slides to introduce, for who are not familiar with this field, what are aptamers. Aptamers can be described by a short linear sequence of oligonucleotides, both DNA or RNA based, In solution, these small molecules can assume a particular 3D configuration as consequence of the binding with a specific target. Aptamers are molecules that are synthesized. So during the synthesis, it's possible to introduce in the chain particular group, chemical group, for increase, for instance, the nuclease, the resistance to nuclease, or to introduce a specific group for their binding to a solid surface. The advantages of aptamers respect, for instance, to antibodies that are the main actors in the biorecognition event is that they are synthesized chemically, so they are highly reproducible and pure, are high stable, they show a high affinity and specificity for the target, but they have a small size and are reusable. The process, the main process which is used to produce aptamers is based on a Selex process. Briefly, starting from a pool of generic oligonucleotide sequence, after the incubation with the target, the sequence that didn't bind the target were discarded, and that are instead binded to the target are amplified by PCR and put again in contact with the target in more strict conditions. So in different buffer, at a different pH, or in complex matrix. At the end of this process that is performed from 10 up to 15 times, generally, you finally have one or few sequences that have a high affinity for the target. What do you have to do next? Well, to bind the aptamers to a solid surface, there are some key parameters that you have to keep in mind. First of all, you have to choose a proper immobilization strategy, selecting the proper chemical route depending from the kind of surface that you have, and choosing between a direct binding on the surface or using an interface. Then you have to ensure a right aptamer conformation using a proper buffer solution, control the distance between aptamer and the surface, and also the density of the aptamers in order to avoid steric hindrance. Finally, you have to passivate your surface in order to avoid a specific adsorption. You have to select the right molecule for this purpose and optimize also the density and check the cross interaction. In the field of plastic optical fiber, there are some configuration in which you can use these fibers. For my colleague already presented a U-bent configuration in which the POF is bended and the core is exposed and covered by gold, or a D-shape configuration, or a fiber grating performed directly on a polymeric optical fiber. In this presentation, I will show you example more related to D-shape configuration. Aptamers, the coupling between aptamers and POF is quite new. If on silica fibers, this coupling started in 2009, in POF started in 2015. There are other biological elements that are usually used to perform a recognition and are historically based on antibody, but also on molecular imprinted polymers that are increasing in their in their use. But for sure, aptamers can be the next biorecognition element. As already told you, we used a D-shape configuration that Dr. Trono well explained before me, so I will skip this part. This is the optical system that we finally use for the main experiment. A first application of this platform was used for the detection of ampicillin, where the aptamer for the ampicillin recognition was directly immobilized on gold through a thiol chemistry, and the optical system was coupled to a potentiostat in order to follow both the electrical signal and the optical ones. The colleague showed the ampicillin detection up to 100 micromolar concentration. But the main application of this platform was related to disease biomarker detection. The first example was based on the recognition of vascular endothelial growth factor that plays a crucial role in the angiogenesis event. On the right, you may see a sketch of the gold functionalization that we use, immobilizing directly the aptamer on the gold surface and passivating with mercaptoethanol. The micrograph here show the fluorescent aptamer immobilization on the surface up to obtain a very uniform coverage of the surface. The transmission spectra show you both the effect of the functionalization before and after the functionalization, showing a red shift of the resonance peak. And in this panel, you can see the detection of VEGF in low nanomolar concentration. The load was estimated to be 0.8 nanomolar. A second example was related to thrombin, that is a clinical marker for blood coagulation cascade and the homeostasis. But in this case, you use a different approach. Respect to the direct immobilization, we decided to introduce an interface based on short peg. This for increase the performance of the system when we will use complex metrics. In fact, in this example, we use an human serum non-diluted, and we check the ability of our system to recognize the thrombin, both using a non-specific aptamer sequence and comparing this result with the previous procedure. The load was always in the nanomolar concentration. A different approach was proposed by Sun using always a D-shape POF, but in this case, over the gold layer, they deposit a graphene film, graphene layer, on which the aptamer was absorbed through the pi-pi interaction, pi stacking. They were able to detect dopamine, that is an important neurotransmitter that can cause neurological disorders when present at low concentration, and they detected dopamine in sub-nanomolar concentration. Another example was based on U-band configuration and was proposed by Sangyai recently. They coupled the aptamer to this configuration that was connected to a smartphone. They used the flash of the smartphone for the light and the camera as the detector. Also in this case, we were able to detect a potential malaria biomarker in the sub-nanomolar concentration, not only in buffer, but also in 20 times diluted human serum. The last example is related to the virus detection performed by our group, we use the same interface layer that we use for thrombin with a specific aptamer for the spike protein recognition of SARS-CoV-2. And we detected the spike protein, purified spike protein, in complex matrix, so human serum diluted 2 times, confirming using non-specific aptamers and aspecific targets. And preliminary results suggest that our platform was able to detect also the positive swap in diluted matrix in physiological solution, suggesting real interesting possibility to approach real samples. In conclusion, the coupling between aptamers and POF device is quite recent, but is for sure destined to increase, also because the production of aptamers is evolved, and not only SELEX process is used, but also system based on molecular dynamic simulation, for instance, the so-called in silico technologies. So probably in the next year, we will observe an increase in aptamer sequence identification. And the main application that are at the moment find are related to the biomarkers detection and virus biomarkers. Here I want to acknowledge all the collaborators that help us to develop these devices, and for sure you for the attention. Thank you.
Thank you, Dr. Pasquardini. Again, another very interesting presentation in the scope of biosensors with optical fibers. Let me check if we have any questions here.
Yes.
Well, I think we can close specifically in your presentation. I thank you very much. And I will take the opportunity to open to everybody the discussions. You can put your questions and answers in the box. There is a specific box for questions and answers. And I'll ask you one question regarding your presentation on aptamer. It's a very interesting because it is a new approach that is being well, new from the last few years, of course. How do you compare this with the other solutions? Do you see some of them being more specific for a specific application, and the antibody, for example, being used for other application? How do you see the future of those two different approaches?
Okay. Well, for sure, antibodies are on the market from very long years. This is the reason why many people are using this kind of molecules, because there is a big viability for many different targets. Not the problem, but of aptamer is that is a quite new technologies. Yes. So there are not so many targets available, and the cost for development the aptamer sequence is high at the moment. First, because the process, the SELEX process, is a long procedure, and it takes months or maybe 1 year to develop an aptamer sequence and to discover aptamer sequence. And this is the main problem. But in my opinion, this new approach that is based on in silico production of of aptamers can reduce the time for sure, and maybe also the cost of their development. So I'm expecting that in the next year, probably we will see the discovery of a new sequence opening very wide range of targets where they can be applied.
But those actually, there is one question which is the same I was going to do. But these aptamers, did you synthesize them yourself, or do you buy them to some specific companies?
No, for the moment, no, because it's quite costly. Actually, I use just the sequence that are already developed, that I find in literature, and I buy it from specific sellers, okay, that they synthesize for us. And there are few companies that perform this work because it's quite long.
Okay, very interesting, very interesting. So as I said, we are now in the questions and answers section, so you can ask any question to any of the presentations. So first of all, I would like to thank Laura, Dr. Laura Pasquardini, for your presentation, and thank you. And we are now going to the open for discussion sections, where everyone can ask questions to everyone. I have one question for all of the three presenters, which is because this is something that I'm quite interested in the sensors applications, but not only from a research perspective, but also how to apply them, how to move them to a high TRL, from taking them from the lab to a commercial application, to a product. So I would like to ask all of the three. I will start with Dr. Cosimo Trono, how do you see this your system being moved to a product? Do you see that short range, long range? What kind of improvements do you see that it can? Do you see ahead of you? Because it's not easy task for this. I know.
Yes.
I did that journey, but it's not easy one. So how do you see it moving forward?
In this case, the aim of these attempts of developing this optical fiber sensor is the simplicity of the system and the portability. So the setup is very, very simple. There is a lamp, an halogen lamp, and a portable spectrometer. And the alignment system is very simple. You don't need to align because you have a big plastic optical fiber in one case, and in the other case, you have a diffusing fiber. So you have to collect enough light to have a signal. So in this sense, I think the implementation of a box with all the setup inside it is simple. From the technological point of view, my experience is in this field. The step is relatively simple because the system is simple. There are not very much issue. Okay, if you have a laser, you have to align and to move, to change the angle, to have a planar movement system. From the technological point of view, this system has several issues. In this case, my opinion is that the system can move simply to an object that can be bought on the market. I mean, this is my opinion, and sorry, I cannot give you information about the market point of view, or this is not my field.
Yeah.
But okay, from the technological point of view, I think that the step is not so big.
Yes, thank you. Yeah, I agree. When you have a simple structure, it's easier to move forward to a different application. And how about you, Dr. Raghavendra Sai? You think it's how do you see your sensor in the next years?
Yeah, in fact, yeah, we do see some issues that will hinder that were hindering us from commercialization. At least I wouldn't talk about commercialization, as you rightly said. We'll talk about TRL levels. That's the best way to put it. So we have been working on this fiber optic sensors almost now completed 10 years at IIT Madras. And before that, I started my training in IIT Bombay. So almost 15 to 20 years, 15 years of experience. So we see that first of all, we have an issue in coupling light. So how easily we can couple light is one of the very fundamental problems if we want to really talk about using these optical fibers in a commercial level, at a commercial level. So other thing is when we sometimes we do see that if the packaging is not correct, and that actually leads to breakage of probes. So especially the U-bend probes. So we have figured out a way to do it, and then we are going ahead with some cartridge design such that these cartridges can be plug and play. So simply just plug it and you should be able to couple light. So we have partly some success, but also we see that the way we have realized automolar detection detection capability, if you want to have the same one for a POC, we need to have a sensitive photo detector. That also, these are challenges but solvable. We probably will need some more time, maybe in 1 or 2 years, before we can solve it and show a demonstration.
Thanks a lot. And now to the last one.
Yes. Okay. So from my point of view, we are quite advantageous because the platform that we use are currently under development by a startup in Milan that is in charge for the industrialization of the platform. So I didn't meet her, this startup, but I'm in contact with them, and that are quite approaching, so in their results. This platform is the platform is quite how can I say, versatile, so you can use with antibodies, with molecular imprinted polymers, for instance. There is a lot of literature made from the Professor Cennamo and Zeni group, for instance, that they prove the ability in recognition very different target. For what concern aptamers, I think that in the next year we will see some products on the market, because there is a lot of interest. Many companies are developing solution based on aptamers, and there is an increase also in the production. So I'm expecting to see this coupling very soon on the market.
Yeah, I fully agree. I fully agree with what you say. So I think we don't have any more questions. So
yeah, Anuj seems to have
sorry?
Yeah, Rogerio, I have a question to Laura, if she can.
Of course, please.
Yeah, actually, Laura and all the speakers, thank you very much for very nice talks, very informative talks. Laura, I saw towards the end of your talk that you talked about the detection of SARS-CoV-2 through your aptamers. Yeah. So I have a very small question in this regard. So are you planning to have aptamers for different variants of SARS-CoV-2? Because maybe that was not talked in that.
Effective, you mean, sir? Okay. It depends from the aptamer sequences that are designed, depend from which part of the spike protein they are designed for, right? So in our case, the aptamer that we are using on the paper, okay, is able to recognize different variants. We have to test it of course. Okay.
All right.
But it depends how you design the molecules.
Sure. So specificity has to be there corresponding to different variants.
Yeah, exactly.
Yeah. Thank you, Laura.
You're welcome.
Okay, so I think we don't have any additional questions.
I just want to give an information to the audience here. So by the way, we actually have started making these U-bend probes by an automated process, and we have been making these U-bend probes using this laser over last 3 years, and whatever published data that we have is actually through that machine. So I'll be looking forward to actually having more collaborations. In fact, what we are doing is silica fiber, not plastic optical fiber POF, but we are using glass fiber, bending it using CO2 laser. So we'll be happy to share our probes if some of the colleagues, some of the friends here would like to actually test them. So
it's very nice. So there is a good opportunity to everyone. So please contact directly because we have this silica bending probes, right? That you develop automatically. I actually have one question. So you do that? Can you do that with different radius, or is it with the same radius?
The core diameter, currently we are using regularly 200 micron fiber, silica, and we are using both silica cladded as well as polymer cladded fibers. We can make 400 U-bend probes with 400 and 600 micron too using the machine. Yeah, but 200 we will be comfortable. We have optimized the system for 200 micron because 200 micron are more sensitive to some extent. They are not so bulky. So that's how we actually prefer 200 micron fibers. So yeah. As far as POF is concerned, we had issues. In fact, nice that Cennamo and the co-workers typically use gold film on fiber. But we are actually trying to do other way. So we are trying to do a labeled assay. So when we are trying to do that, we do see some functionalization problems with the POF, and they are not as good as silica fibers. So otherwise, we in fact wanted to actually go full onto the POF, but we couldn't go really because the functionalization issues are there, and that's how somehow the sensitivity that we achieve with silica fibers, U-bend fibers, we are not able to do that with plastic optical fibers. So this is one of the major issues we actually want to try. And if there are any suggestions, definitely we'll be very happy to take the suggestions.
Okay, thank you. Thank you once again. So, I'd like to thank to all of the three speakers that give us a very nice presentation, all the explanations that we have made. It was great to see these developments that are being made on the optical fiber sensing, specifically for these different applications. And we have so thank you everyone. We will move to the next section, but to be right on time, I will ask you for a 2-3 minutes short break, and we will start at 9:10. Okay, we are back. Thank you for waiting for these 2 minutes. So now we will continue for the second part of this session. The quality, I believe, will be at the same high quality as last presentation, and we will start with Dr. Anuj Sharma from National Institute of Technology in Delhi. With the title will be Plasmonic Sensors and Detectors. Thank you.
So hello everyone, and it has been really a very nice time to listen to all of your talks so far. And my name is Anuj Sharma, and I'm working at the National Institute of Technology, Delhi, India. And as you can see, the topic of my talk is very straightforward, very simple, plasmonic sensors. So actually, my group has been working on slightly different aspect related to plasmonic sensors. We usually work on the performance enhancement techniques. So it of course contains different improvisations in plasmonic sensors probes. So I'll be discussing them in brief. Of course, not all of them can be covered in 15 minutes first, but I'll try to discuss a few of them, very important ones of them. So usually we work on plasmonic sensors and plasmonic photodetectors, utilizing the plasmonic excitation. So during this talk, I would like to take you through a few things like the fundamental concept of optical sensing and plasmonic excitation for sensing. Then, of course, there have been several talks on optical fibers in SPR. I would like to touch upon that also. And then in the last half, I would like to discuss the role of different two-dimensional materials like graphene and MOS2, etc. And recently, we have also worked on a very interesting aspect, which is the manipulation of radiation damping in plasmonic sensors. So that will be another discussion on that. And I would conclude with some focus areas for the future research. So if we talk about the optical sensors, of course, if we basically define it, it is a technique involving the light modulation for recording constant parameter. And of course, it has three parts, as my cursor might be visible to all of you, that we have a light source, and we have a detector unit here at the end. And in between, we have a combination of a coupling device and the sensing technique, of course. So these coupling devices can be optical fiber in most of the cases. Then we can also have prisms of different glass materials. So that is another important area that we have focused on. We can utilize the prisms of silica, chalcogenide glasses, even in some cases silicon also. This coupling device can be used in planar waveguide forms also. Okay, so if we say that the modulated light, I mean, this is the modulated light which has passed through this coupling device and the sensing region. It carries the information about the properties of sensing region. So whatever is in the vicinity of this sensing region, that will be correlated to this modulated signal. If we look at the various factors that are involved with the optical sensors performance, so we should focus on sensitivity. Of course, Professor Sai has been talking about it in his talk also, that the sensitivity should be as high as possible. So of course, it is the capability of comfortably distinguishing in different analytes. Okay. Then we can also focus on accuracy, how precise our measurement is. There should be minimal errors. And of course, in case of SPR-based sensors, this becomes a very important feature. And when it comes to the practical realization of the sensors, we should have as small as possible response time. Operating range should be as large as possible. So the sensor probe should be able to detect a very large range of analyte parameters, be it refractive index, be it some bioparameter, be it temperature, etc. And detection limit, of course, it should be very small, which is a very important feature. Now, as I mentioned, that there are several techniques available. For example, we have evanescent wave absorption spectroscopy. There are more fluorescence, etc. Many techniques are available, but it has been SPR, which has been at the helm of the techniques available in the last 3 or 4 decades. So yes, SPR is a very nice and very interesting phenomenon, and which has still a lot of possibilities in terms of the performance enhancement, as well as, of course, as all of you mentioned, the product based research. So if we just define plasmons for the general audience, these plasmons are nothing but the coherent oscillations of the free electrons on the metal boundaries, which is a concept which we study in our graduate classes, of course. But when we combine this metal with the dielectric, then these plasmons are found at the interface of the two, and they are known as the surface plasmons. The surface plasmon oscillations are highly coherent, and they have a longitudinal electric field associated with them. In general, if in the language of optics, the surface plasmons are TM polarized or bipolarized. Well, how can one use the SPR concept in sensors? So Professor Trono was talking about the Kretschmann configuration. It has been, of course, one of the most popular ones. Kretschmann configuration allows the coupling of these surface plasmons at the metal and dielectric interface with the evanescent wave generated through the total internal reflection or attenuated total reflection when we launch the light through a prism. So this is one of the basic geometries we have. And of course, when this happens, then this modulation can be measured in terms of the reflected beam. And of course, the reflectivity encounters a very sharp decrease when the resonance takes place. When the properties of this evanescent wave and surface plasmon wave match with each other, then we have a sharp resonance, which is actually represented by a simple equation also in terms of the propagation constant or the wave vector matching. So, but the important part here, apart from this, is that this dip will be at different locations in terms of the incident angle or in terms of incident wavelength also in many of the cases. And this shift is basically the representative of the sensing feature of SPR. So yes, as I just mentioned a couple of slides back, that for different techniques we have different parameters or different benchmarks for the performance evaluation. Well, in terms of the SPR sensor, if we talk about a simple this spectrum of reflectivity, there can be 2 aspects, of course. One is the sensitivity aspect and another one is accuracy aspect. Okay, so sensitivity aspect is in the terms of the shifting of the curve. So as soon as you have a very small change in the analyte property in terms of any parameter, then it should shift to as large extent as possible on this scale. And accuracy is basically related to the sharpness of the curve. Okay, so I was just looking at a few SPR curves in one of the presentations. Well, yes, the width was very crucial, because if you have a sharp SPR curve, then the accuracy will be naturally large. Or if you have broad one, then the error here in measuring the resonance angle or resonance wavelength will be large, and it will basically hamper the accuracy. So yes, Rogerio was asking about the diameter or the radius of the fiber. So when you ask a theoretician or you ask a person who is working in performance enhancement techniques, it is a very important parameter to tune the performance. So of course, in terms of the prism-based geometry, you have the refractive index of the prism means material, which material you are talking about. Then you have metal parameters available, thickness, roughness, and the choice. Of course, not all the metals have SPR properties. For example, silver and gold are found to be good SPR active metals, but not others. Then you have external parameters also affecting the performance, like temperature, humidity, etc. And then there are others, other parameters or other factors also, like which sensing medium you are talking about. Is it an absorptive analyte, or is it just a plain analyte? Then you have buffer layers, you have connecting layers, and the wavelength, of course. So that there is definitely a lot of possibility there. So when you talk about the selection criteria of wavelength region for sensing, yes, there are multiple options available for us, depending on the analyte, depending on which application you want to have. Of course, when you talk about the chemical and biological species, they have their fingerprints in IR region mainly. They have good absorption there, so maybe near infrared or mid infrared can be preferred. But then it makes the sensor design a little bit more complex. So yes, then if you go for the shorter wavelengths, like visible ones, then the photo damage will be something which we have to take into account. Then the connection between the wavelength and the Rayleigh scattering loss will be there. So there are different considerations, and one need to be very specific and very careful while selecting the wavelength. And of course, a lot has been already said about the optical fiber sensors. So I would like to just touch upon it very quickly. Yes, prism-based geometry can be replaced with a fiber-based geometry, and it is basically a very good choice, because the flexibility and the robustness of the optical fiber systems is something which is not available with the prism-based and the planar waveguide-based designs. So, of course, in addition to what we had discussed in the last slide, there are more parameters to tune. Like I was mentioning that the diameter of the fiber, length of the fiber you are using, numerical aperture of the fiber, etc. Well, length is also very important. I would like to highlight slightly this factor, because when you take the large length of the fiber for remote sensing applications, let us say, then it affects the overall power distribution also. There have been some very pioneering works from some very prominent researchers, and they discussed the effect of the length of the fiber on power distribution. And in case of SPR sensors, it will basically affect the power, the sensor performance prominently. We have had some work on that in the past few years, and we have got some very nice results, and we try to optimize what kind of length, what kind of diameter should be there to find out the best performance out of the sensor. Well, these are a few possible geometries with the optical fibers, which is a precursor for different biomedical applications. So you can have a terminal SPR sensor. These are all fibers. When we see these are the fibers. You have a reference fiber for taking the relation with the signal fiber, and then you can finally have a spectrum somewhere in the OSA. Well, yes, we have had some very interesting work with fiber optic SPR sensors. For example, we proposed a design for blood group detection by utilizing the dispersion characteristics of different blood groups at some particular wavelength. Sorry, not at the particular wavelength, but at the range of the wavelength. So we tried to propose a design of this SPR-based fiber optic probe for blood group detection. And we also try to maintain the specificity, like I was asking Laura about that. It is very important in terms of any biosensor that the specificity or selectivity should be maintained. So you need to use appropriate materials. Like here, buffer layer has been put in the design, which can be an example like polyurethane or some thiol layers which can be used. But yes, we need to have very correct relations for those layers. Then the design parameters can be optimized. Of course, this is just a simple example I'm giving for the sake of simplicity of understanding. but optimization is done in very small steps and a very high accuracy is required. Well, yes, the outcome is on the similar lines, that we have resonance spectrum, the SPR dip at different wavelengths for different blood groups. Of course, it is once again a note of caution that one has to be very, very careful with the specificity. We did some simulation work, and which got some recognition at that point of time also. And yes, there are more applications of plasmonic biosensors and chemical sensors. Plenty of examples we'll find in literature. There are some examples here. All right, so now I have moved to the second half of my talk. Of course, maybe I'm left with 5 minutes only, I think. So the role of two-dimensional materials we have been focusing on in this performance enhancement for last few years. And yes, there are many examples of two-dimensional materials we have. Well, one of the most famous ones is graphene of very sub-nanometer thickness, 0.34. Generally, it's not necessary that it is always 0.34 nanometer. It can be in this vicinity itself. Then we have some dichalcogenides also, like MOS2, MOSE2, etc. These are a few examples only. We have several heterostructures available these days, which are active in different wavelength regions. So, of course, those who are working on different biosensors, this can be another important and interesting addition to their work. So they have several good advantages which can be explored in SPR sensing. Like for example, we worked on utilizing the variation in the chemical potential or variation in the doping of graphene layers for enhancing the sensor performance. And we found that the refractive index of graphene, of course, it's real as well as the imaginary part, or let us say its reflection as well as the absorption properties are heavily dependent on chemical potential of graphene. These results are at 1.55 micrometer wavelength, and we utilized, we translated this variation into the SPP penetration depth at this wavelength, and we have some very interesting results. On this vertical axis, we have the variation of combined sensitivity factor, which takes both sensitivity as well as accuracy aspects into account. And we did the simulation. This is just one result for your reference. We simulated this for a different number of graphene layers, and we have some very interesting results. Of course, we can go up to some good levels of performance, but yes, maintaining the chemical potential or the doping of the graphene layer is a very cumbersome task, but literature have confirmed that the chemical potential can be varied up to 0.6 or 0.65 electron volts by utilizing the electrical gating or chemical doping methods. Well, later on, I mean, for last 1 or 2 years, we have been working on another very important technique, which is the manipulation of radiation damping. This slide will give you that flavor that what kind of concept we are talking about. Radiation damping is basically depends on the nature of interference between the incoming light and the backscattered light. So this is just one very simple diagram I have put, that this is the thin layer of metal, and we put we incident some light on that of any given wavelength, and the thickness of this thin layer is dm, let us say. So this dm and lambda combination will be very important in deciding the overall dip behavior. Okay, so we tried to exploit this this concept of destructive interference between the incoming and backscatter light that leads to this zero reflectivity. So in effect, it is a phase variation phenomenon. This is just core physics. And when we talk about the tunability or the manipulation, then we have to basically find out or look out for those combinations of lambda and dm, which gives us nice performance and very high values of the figure of merit or let us say sensitivity. So we tried to do some very high accuracy simulations on that, and we got some very nice results that this FOM, this 2D variation is representing the FOM, and this figure of merit or FOM, let us say, has gone to a very high level of magnitude for these kinds of combinations. Yes, lambda can be got with some specific light sources, but finding out this kind of thickness is not an easy task. So of course we are improvising more on that. We will try to find more related combinations so that practical viability is there. But these are some very encouraging results. Some more results, I'm not going into them, and some more explanations have been given. And then we also tried to exploit another combination by utilizing the graphene and alumina heterojunction. Well, yes, we also used it in solar cells in some separate works, but here in plasmonic excitation, this has also been used. Of course, for these also, we have some very prominent results, very interesting results, and the FOM can go up to values of, I mean, these are, these may look absurd, but we are working on them right now and trying to find out, as much practical viability as possible. But simulationally speaking, these are very encouraging and motivating results for us. Well, before I conclude, I would like to just touch upon the focus areas for future research. We are working on the corrugated structures in plasmonic devices, especially on sensors and photodetectors, and we have had some very good success, both practically as well as theoretically, and we have already published some good papers, and we are in the process of some patents also. Then 2D materials, as I just said, they are still a lot of scope there. Then the introduction of new concepts in plasmonic devices. For example, I'm running a project these days sponsored by an Indian agency, which talks about the role of spin waves or spin Hall effect and magnonics in plasmonic devices, and we are getting some very nice results. The project has just started, so let us see if it gives us some very interesting results. And of course, it goes without saying that FBG and PCF based plasmonic devices still have a lot to do. Polymer fibers, as many of you have already talked about. Then the terahertz plasmonics is also a very upcoming area. A lot of research has already been done, but still a lot of scope is there. Well, thank you, and I would like to thank my collaborators, my funding agencies, my colleagues, friends, family members, and of course all of you to give me this opportunity to be here today. Thank you very much. This is a photograph of our upcoming campus in Delhi. We are right now in a rented campus, but in a matter of 6 months or so, we'll be in our own campus, and this is just a very nice picture of that. Thank you very much. Thank you very much.
Thank you, Anuj. Thank you very much. It will be a nice campus to move on next.
Thank you. Thank you so much.
Okay, so we are a bit delayed here. So if you don't mind, we will have all of the questions in the end of the discussions. Okay, thank you. Thanks a lot. So we will move to the next speaker, Dr. Christophe Caucheteur from University of Mons in Belgium. Christophe,
yes,
the floor is yours.
Okay, thank you very much again for this very nice introduction and this invitation. It's a pleasure for me to present our work on plasmonic fiber grating biosensors. I'm very happy to see all the previous presentations showing extremely nice results, and indeed as well relevant information for this talk, because we all aim finally at transposing the Kretschmann prism into an optical fiber counterpart. And in our work, we rely on the telecommunication grade single mode optical fiber, in which we engrave a fiber grating. So this is where we are different from colleagues in the field. Obviously, the fact that we produce it in an optical fiber is to gain a very weak intrusion and to make in situ measurement possible. And indeed interrogate remotely a very small volume of analyte. We talk typically about microliter volume or even less, and provide real-time measurement. So in our case, we use near-infrared light, so really around the telecommunication window at 15 nanometers. And we rely on grating that behave as spectral combs, as I will show you in the next slides. Indeed, finally, when you look at the modes of an optical fiber, our probes, they are among the only ones able to excite simultaneously but distinctively all these modes. And this is where we can have a high sensitivity because we rely on a narrow band resonance, as it was shown before by Anuj. So the research context of our work is to provide a probe for cancer diagnosis. And if we talk about cancer diagnosis, the idea is to do it in vivo, typically in areas of critical access, like the lung, for instance. We are working in this field since about 10 years now. At the beginning, we were supported by an ERC grant, and now we are supported by an Excellence of Science grant in Belgium. By the past, so the focus was on lung cancer, and now we focus on two relevant biomarkers for breast cancer, the mammaglobins, which are cells, and a specific protein, the human epidermal growth factor receptor, so HER2, and I will show you our results there. In the bottom of the slide, you can see an artist view of our probe, finally, which contains different parts, and I'm going in the remaining of the presentation to discuss about these different parts. So I will give you some word about plasmonics, but everything has been introduced so far, and so I will be extremely fast there. Then show you what kind of gratings we use and how we produce them. Then give you some words about the gold coating process, but it's a classical one and very similar to what colleagues use. Then give you some words again on the biofunctionalization process and show you our results obtained in different bioassays. So, well, no need to recall what is a surface plasmon resonance, I guess. So there are some definitions here, they are just for memory. Just to say once again that indeed we want to transpose the Kretschmann prism configuration into an optical fiber counterpart. So in the Kretschmann prism, light is launched with an angle beyond the critical angle and is just totally internally reflected at the gold surface, so at the flat surface where a thin gold layer has been deposited. This produces an evanescent wave that extends in the medium typically of half of the wavelength or even less. And so, of course, the power decays exponentially. But this evanescent wave, upon certain conditions, can excite a plasmon wave at the interface between the metal and the external dielectric medium. And this gives a resonance. And you have understood that this resonance will shift in response to a change of the surrounding medium. And so from the blue curve, you get the red one. And obviously, by looking at this shift, you can know that something has happened at the surface of the sensor. So it is, of course, used to measure density fluctuation. It is used to measure thickness changes. But probably the most use you have of that is, of course, when we rely on molecular adsorption and when we implement like this a biosensor. In our work, I said already, we rely on the glass silica, on the glass fiber, I mean the one of the internet. And so it's a single mode fiber. And in the core of the fiber, we produce a grating. So it's a periodic modulation of the refractive index along the fiber core. And instead of using uniform grating, so the very well-known use in telecommunications, we use slightly tilted fiber Bragg gratings. So you can see here that the modulation with respect to the perpendicular to the optical fiber axis is slightly angled. And as such, we get this transmission spectrum. So you see here the transmission spectrum is composed of several tens of cladding mode resonances below the Bragg resonance that corresponds to the core mode coupling. So the Bragg resonance that is there, it corresponds to light that remains guided in the core, and it can be used as a thermometer finally. So it's your temperature reference. And when you use the other cladding mode resonances, this is where that you can probe extremely finely the refractive index of the surrounding medium. And depending on where you are, you will probe a different mode, as you see. So typically, when we talk about the refractive index of water, let's say around 1.32, you will use these modes that are located here around the wavelengths of 15, 40 nanometers, okay? So among the comp, we really select the modes that are the most sensitive. Then we can also rely on so-called eccentric gratings. These are gratings that are produced close to the core cladding interface. So they are no longer centered in the core, they are close to the core cladding interface. And you see typically here the spectrum that we get from that. You see that it is a very high spectral comps that can finally extend to reach the refractive index of air. It's important to mention that in the left part of the graph, the effective index of the mode is close to 1, the effective index of air. In both cases, for tilted or eccentric grating, you break the cylindrical symmetry of the optical fiber, and in that sense, you will be able to generate a plasmon wave because there will be a very nice polarization dependence. And upon the right polarization, the plasmonic generation is possible. But now you could ask how to produce these two gratings. Well, it's extremely simple. For a tilted grating, we will rely on the phase mask technique. You see here on the top figure an example of a phase mask, and the phase mask is slightly tilted in the plane perpendicular to the incident laser beam. We can have access to different lasers in Mons to produce that, an excimer one, a continuous one, also a femtosecond pulses laser. For the femtosecond pulses, it's important to mention that when you use a microscope objective and you do the point-by-point writing process, this is where you can achieve the production of eccentric fiber Bragg gratings. So it's quite simple. And now what happens when you put the grating, the bare grating, in a solution and you change the refractive index? Look, this is what you can see here. So you see spectra. And if you now zoom on the mode of the most sensitivity, you see that when you slightly change the refractive index, so you change from 137.28 to 137.70 in this case, you get a progressive shift of the cladding mode resonance. And if you now track this shift as a function of the surrounding medium, you get a linear relationship with a sensitivity typically of 20 nanometers per RIU. Knowing that for this particular case of grating, we rely on devices that allows a wavelength resolution of 1 picometer. We are extremely precise in the demodulation. So this is for a bare grating. And then we put gold on this bare grating using typically the sputtering, as it was mentioned before. You can see here an SEM picture of the gold layer, just to show you that it's of relatively well uniform all along the fiber cross section. You see typically the chamber we use for the sputtering, and the gold target is on top, of course. And now that we have a gold coated grating, depending on the polarization that we inject inside the structure, we can excite the plasmon wave. This is typically the example of the red curve, where the electric field is perpendicular to the metal. It corresponds to the P polarization mode. In that sense, the excitation of the plasmon wave is possible. If you come with the orthogonal state of polarization, then the electrical field is such that it cannot enter into the metal, and you will not be able to generate the plasmon wave. So the message of this slide is that with the right polarization, we can generate the plasmon wave. When we have that, we now have to focus on that particular wavelength, you see, just below the signature of the plasmon wave. And again, tracking the shift, we get a linear relationship, but you see that this time the sensitivity has increased compared to a bare grating. We talk now about 50 nanometer RIU. We have examples where it is even higher because it depends also on the gold thickness that you place. Okay, now we have the grating, we have the gold coating around, and we generate a surface plasmon wave. And now the next step is to biofunctionalize this platform, of course. In our work, we rely on the two types of bioreceptors, antibodies and aptamers. So aptamers, they have been very well presented before, and I just have to emphasize here that they really produce a high affinity with a very high specificity, and this can be indeed synthesized using the SILEX method, as you have seen before. The antibodies, they are pretty much the same, a bit bigger in size, but they also offer high affinity and high specificity. And so we have both. And in both cases, the functionalization on gold is a double step process, I would say, because we first need to attach a self-assembly monolayer so that we can graft the biosensor on gold, the bioreceptor on gold, sorry. In the particular case of aptamers, they first of all have to be reduced, and then they could be immobilized. I have to say here, frankly speaking, that I'm not at all a specialist in this field, and in my team, there are biochemist colleagues who are making this process for us, because my background is in photonics, as you know, and I'm not at all an expert. But then I wanted to show you that with this probe and its functionalization, we can target cells or proteins, and these are not at all the same size in terms of analytes. If we talk about a cell, we talk about something typically of micrometer scale. If we talk about a protein, it's extremely small. And so these are not the same, but it will work with the probe. Here we see an example where we track the mode of maximum sensitivity as a function of the process that we make around the optical fiber. So we start in PBS. Then here it's the biofunctionalization process that is followed. So it's the aptamer immobilization. Then we have the surface passivation. And then starting from that point, the sensor is ready. And now we will use it in a control solution. And you see that apart from a very small drift, nothing happens. And then we bring the sensor into the target solution containing here 100 cells per milliliter, and the cells is the mama globin in this case. And you can see that in this specific case, the response rises and we are able to detect these cells. Okay, so this is one example where we see the surface activation and then the use of the sensor. Another example here, just to show you the possibility to detect the HER2 protein, where we first have the sensor in PBS, then we have the protein, and you see that again we have a very nice response. Then we come back to PBS, and then we make an amplification of the response using a gold nanoparticle on top, and you see that it rises again and then back again into PBS. Okay, if we look to the specific signature that we track in this case, you see we have a zoom in the spectrum there showing the point that we track and how it goes for the demodulation. Right, so I said at the beginning, our wish here is really to go towards in vivo diagnosis. In vivo means we will use an endoscope and enter the human body with our sensors. And so we have already placed the sensors in a catheter, a specific one. You see here close to €1 coin. You see how small it can be. And this catheter can enter the operating channel of an endoscope. So you see there the example. The catheter was made in such a way that you have 2 stainless steel blocks and that it can be seen under X-ray spectroscopy. And so far it was tested in ex vivo tissues. So we were at the hospital with our system. You can see the readout device here. You can see the signature and then the sensor in the tumor here to discriminate healthy and tumorous tissue. So we have worked ex vivo with human tissue, and we have even worked in vivo in an animal system. So here it was a pig, and we were able to bring the sensor into the pig lung and to make some measurements there. Of course, the pig has no cancer, so we were not able in this case to measure the presence of a cancer, but just to show that we have the sensor operational within a living body. And I think that it's quite very relevant for the moment. So this brings me already to the conclusion. So I hope I was able to confirm that our probe is a low loss system that is able to finally look at molecular interactions, that it can be produced quite easily with standard single mode optical fiber and at any wavelengths. It has a limit of detection that is suited for much target applications you can have, and specifically in cancer diagnosis. And since almost 10 years now, my team has really accumulated a lot of experimental evidence about the repeatability and high sensitivity of our platforms, both in vitro, ex vivo, and even in vivo, as you have understood. Thank you very much for your attention.
Thank you very much, Christophe. Indeed, a very nice presentation about the developments of your group in this topic. I have some questions, but since we are a bit delayed, I will save all of the questions from everyone for the open discussion. Okay, thank you very much. Okay, so moving to the last but not the least presentation, it will be given by Dr. Robert Meier from Precision Sensing in Germany. And the title of the presentation you can see already in the presentation, which is Fluorescence Optical Chemical Sensors, Past, Current, and Future Perspectives. So please, Robert.
Thank you. Thank you for the kind introduction. Yes, you can already see my title. Actually, our company understands the fluorescence optical chemical sensors in a way of oxygen, pH, and carbon dioxide, and also lately biomass sensors. As already said, I'm from a company which is based in Regensburg in the south of Germany. We've been doing this for approximately 25 years by now. We started as a spin-off from the local University of Regensburg and are specialized on monitoring oxygen and pH and CO2. Currently, we are more than 100 employees, and we offer certified quality according to ISO standards 9001 and 13.5, which is the standard for medical parts. Oxygen and pH and CO2 play a key role in metabolism and a lot of other applications that are very useful to understand for life or for understanding your processes. And I hope that I will show you some applications where these sensors can be applied. First, let's start with the basic principle of our sensors. Actually, our sensors are based on luminescence, that can be fluorescence or phosphorescence. These dyes, or usually you have molecules that show an effect called luminescence, where you can excite them with a specific wavelength of light, and then they show an excitation. Actually, very many of those dyes are available in the market and also in the scientific and in the knowledge, but only some of them show special properties like reversibly reacting on a specific analyte. Actually, when I'm talking about past, let's do just a quick look what happened during the last century. Actually, in 1977, the first pH-dependent fluorescence spectra of an umbiliferone was reported by Professor Otto Wolfweis, followed by a fast-responding fluorescence sensor for oxygen detection in 1984, and a first triple sensor for measuring oxygen, pH, and carbon dioxide in 1994. And in 1997, our company was founded. Just as an example, one of the principles that we use is called dynamic quenching of specific dyes or specific fluorescent dyes. The situation can be described simply in case of these two situations. First situation is we have the plain dye, we shine light at it, it gets into an electronically excited state, and then is able to send out a redshifted fluorescent response. And some of these specific dyes have the properties that they can interact with specific molecules. For example, in this case, with an oxygen molecule. So in presence of oxygen, the oxygen molecule can bounce against the excited state fluorophore, transfer the energy to the oxygen molecule, get back to its non-excited ground state without sending out any fluorescent response. And the oxygen molecule sends out the response in a non-visible spectra emission. So is such kind of a dye yet a sensor? Actually, no. So a sensor is always a full system. So we need something that has an effect, so that reacts on a specific analyte. Then we need to generate this effect, for example, by shining light at sensors, and then have this photophysically interaction. Then we need a referenced readout of the effect, need to translate the effect into an analyte signal, and indeed generate an output so that the user can use this information for interpreting his system. And this whole system is then officially called a sensor. So in our case, we mainly have the sensing dyes incorporated in such kind of small patches, which can be attached to a vessel wall. And then these fluorescent reporter dyes in there translate an invisible sample component, in this case oxygen, into a light signal. So we excite the dyes with a specific excitation light. It generates a redshifted fluorescent signal. And this fluorescence response has the encoded quantitative information about the respective analyte in it. And the signals that are generated are recorded by a measurement device in a reference manner. And the measurement device itself processes the data and then gives you, for example, an oxygen value of the situation in your sample. The benefits of these optical chemical sensors are, first of all, as we use light as signal transducer, you can measure through optically transparent materials such as glass walls, plastic walls, or vessel walls. Thereby, you can keep your system closed. You do not need to open it. You do not need to do sampling. So you can avoid, for example, oxygen ingress in your system or CO2 emissions from the system. Second point is these patches are not made out of glass, so there's no risk of breaking. So they are pretty easy to handle. And compared to electrodes, these materials do not consume the analyte. So it's not a reaction, it's a photophysical interaction, and it's totally reversible. This also has the advantage that the sensors can be used independently from the flow velocity. That means you can use them in static systems, but you can also use them in flow systems. And another advantage is that they do not show or they are not prone to electrical interferences. Actually, our customers or our users have a pretty broad range. Maybe the main application field is biotech and pharma, where these sensors are usually used to monitor bioprocesses or to monitor how your cell culture or your culture grows, if it feels well, and if you need to adapt something in the process, for example. But they're also widely used in food and beverage applications, for example, to test whether a protection gas atmosphere is still valid, or if the material is able to retain the protection atmosphere, or if your production process is still in good shape. Further on, they're also used in biology and environmental applications, and in industry and technical applications. And last but not least, our sensors are also used in medical and life science applications. For example, we deliver components for minimally invasive brain sensor or for blood perfusion monitoring. So how do these sensors look like? Actually, we offer different kinds of application formats of these sensors. The simplest ones are the so-called non-invasive sensor spots, but we also offer them as plain sensor sheets for generating 2D information, as micro-invasive sensors, and as turnkey solutions where you have specialized sensor systems covering one specific application. So let's talk about a little bit about our mini sensors, or the so-called sensor spots. Actually, these kind of patches, they can be easily implemented into nearly any kind of transparent vessel. You just stick them on the inner wall, and then you're able to read out the analyte content, maybe oxygen, pH, or CO2 on the inside from the outside, just by using the light signals to transduce the signal. This is a non-invasive measurement technique, so you can keep everything closed. You do not interrupt your sample. You can do sampling-free real-time monitoring, and another time, but it's pretty important. They do not consume analyte and are non-toxic, so you can use them, for example, in bioprocess. This is maybe the original format of our sensors, the so-called microsensors or probes. In here, the sensing chemistry, so the fluorescent components or sensor components, are immobilized on the tip of a fiber. It can be a glass fiber, or it can be a polymer optical fiber. And then you can use these for minimally invasive measurements, or also for robust applications where you just need to put in a probe into a semi-solid sample. These are mostly used for profiling in biofilms and sediments, for insertion in tissue, or also for measuring in very small volumes, for making micro-respiration systems to see, for example, in only a few microliters, how a component consumes up oxygen, or how the pH changes, or for intruding them into packages without harming the package total constitution. And they also offer a high spatial resolution of below 50 microns. Actually, this is the topic I'm mainly working in. I'm a 2D guy, so I'm working in the imaging department. These sensors can also be in the format that you have a planar sensor sheet, which is then recorded with the camera system. And thereby you can generate more than 100,000 sensing points within one single image. That means you can gain 2D information, actually generating analyte maps over your sample. You can also record that over time, so you can generate videos of that, and you can see where in your system what happens. And then you can also use this data for analyzing it further by, for example, comparing different regions of interest, or by analyzing gradients over interfaces, or over whatever your sample forms a gradient with. As last application format, we also offer turnkey solutions for specific situations or specific applications. These are always packages, system packages that contain a software, a meter, and some disposables that are specialized on specific application formats. For example, like measuring in well plates in the incubator, or measuring on a shaker, doing parallel measurements. They are always non-invasive, non-destructive measurements, and the disposables are pre-calibrated and ready to use. And these sensors are available indeed for oxygen, pH, and also lately biomass and CO2. So what comes next in the field of optical chemical sensors for oxygen, pH, and CO2? That's a good question. I personally believe, actually, I have to, because I'm working in the imaging department, but I think there's a big perspective with terms of detecting multiple sensors in one scenery, just with camera devices, combining different sensors, combining different informations, etc. So thank you for your attention. And if you have any questions, you can ask now, or you can also just write me a quick email. Thank you.
Thank you, Robert. Thanks a lot for the presentation. Again, three very good presentations that are in different fields. So it is this session has been very good in terms of how the evolution of the optical sensors for different chemical analysis and biological analysis. We are now entering again the question and answers. If anyone has any questions, please feel free to put on the question and answers box. Actually, I can already start with you, Robert, that you just finished your presentation. And you mentioned that you can measure the pH, the oxygen, and the CO2. I wonder the measurement that you do. The oxygen, it's in the liquid, so it's dissolved oxygen, or how is that?
Oxygen sensors work in both environments, liquid and aqueous and gaseous form. So you can also apply them for measuring atmospheric oxygen, for example.
Yeah, atmospheric, yeah, but in liquid, how is that done? So this is a dissolved oxygen that you measure?
Mm-hmm. Yes, so actually the sensor is incorporated in some kind of a polymeric film, and the whole principle is diffusion based. That means oxygen molecules can diffuse from the liquid or from the air phase into the polymeric phase, there undergo the photophysically interaction with the sensing molecules and generate the signal. So actually the oxygen sensor does not care if there's liquid on the other side or if there's gas on the other side, as long as there's some diffusion processes going on.
Okay, as long as there is molecules around that can diffuse, okay. And the other question is, have you considered or have you done before the measurement of sulfur dioxide?
I personally not. I'm also not aware if there are responsive fluorescent materials that specifically interact on sulfuric oxide, because if you want to make an optical chemical sensor for a specific analyte, you need to be sure that it reacts only on that specific analyte and that you minimize the cross interactions with other components. You can do that usually by shielding it with specific matrix materials. But I'm not aware if there's an sulfuric oxide sensor on the market or in literature yet.
Okay, thanks. And also for the pH sensor, I was wondering about the reproducibility of that. So you can, how accurate can you be with the measurement of pH in this kind of measurement?
So the pH sensors have one big drawback and one big advantage. They do not have the broad range compared to the electrodes, because with an electrode you can go from 0 to 14, for example. The fluorescent sensors, usually they have a response of 2 to 3 units of pH, but in that range they are pretty sensitive. That means that usually with our sensors you can gain 0.02 pH units precision.
Wow, it's very impressive. Okay, thank you. And now I have, let me check if you have any additional questions. I don't want to monopolize all of the questions here. Okay, and I have, from my side at least, I have one last question. It's from to Christophe, that you showed the in vivo and the ex vivo applications of the probe. I was curious about the probe that he used. It was really small, and the tip of the fiber is how do you put that in contact with the tissue? Do you have any?
Right. So we develop a specific catheter. The size of the catheter is 2.2 millimeters because it enters very well the operating channel of the endoscope. So it's a catheter in Teflon that contains a window, and in the window we have the telecommunication grade optical fiber. So it's a single mode fiber that is placed between two blocks in stainless steel, as you have seen. And so we have a lateral contact between the sensor and the tissue. And it works very well like that, indeed. So it's quite robust, finally. And of course, we rely on the robustness of the optical fiber. You know that for that particular case, the robustness is there. And so it works pretty well, indeed.
Okay, thanks. So it's a lateral contact. Okay.
Yeah, lateral contact, yeah.
So, okay, very nice. Thank you.
Thank you. Thank you.
So we are also arriving to the end of the session. Any more questions from anyone? Any comments?
Coming to Christophe's work, so I just have one query there. We actually have gratings based sensors separately, and then SPR based sensors separately, and then you actually have combined it, putting together. So is it that you're because of plasmonic effect, you're actually getting more reliable and then more sensitive response from these sensors, or you're using it for many other reasons like functionalization and other things as well?
So from the sensor in itself, you mean?
Sorry?
You mean from the sensor point of view in itself, comparing a multi-mode configuration finally, where you get a broad response, and our grating where you get a narrow cladding mode resonance. This is what you mean?
Right. So what I meant is we have FBG kind of gratings. So they themselves are standalone sensors.
Yes.
And we have plasmonic sensors. They themselves are standalone sensors.
Yeah.
So basically, you have combined these two.
We can
very
combine
nicely.
it. Yeah.
Very nicely, you have combined it. So the advantages that you have are you have a very high improvement in sensitivity, or
Yes.
any other advantages that you are actually having by this?
Well, it's essentially this one. So the other advantage is that we use all equipment devoted to the telecommunications. So, I mean, we are using the classical source and detectors that are there for FBG in telecommunication as well. So it's clear that we can see it as an added value. For processing the fiber, it's the single mode fiber. So we use a standard stripper, standard cleaver, everything that is standard. So this is the other point. Now we have been looking at benchmarking these devices. So the gratings compared to the classical, let's say, unclad fiber approach, where you have typically the multi-mode fiber, 400 micrometer, for instance, in diameter, and that is unclad, and you put the gold coating around. There, you have this very broad resonance, and this broad resonance, you can only measure it with a spectrometer whose intrinsic performance is limited, of course. But okay, this is extremely straightforward approach and extremely interesting in practice. Looking at the limit of detection, we believe that most of the time, the limit of detection of such devices lies in the nanomolar range, something like that, nanomolar. And I would say it's the intrinsic sensitivity of the unclad configuration. With the grating, we believe that we can reach intrinsically picomolar detection. Then your work is essentially based on decreasing this further, and you have achieved attomolar. This is extremely tough to get there, of course, because of the extremely low concentration. It's even lower than what you can get with a spectrometer approach. So it's clear that there is really research oriented towards decreasing as much as we can the limit of detection. But okay, we remain intrinsically around the picomolar. So
yeah, but the point was actually not really comparing multi-mode fibers, but there have been many studies. Professor Formula, he has pioneered single mode fibers and SPR based single mode fiber based sensors. So so what I just wanted to ask was, basically you have very narrow shifts, so that's how you are going for this, and of course this is, you are able to tune the resonance wavelength at will, right? Okay.
Yeah, this is it. We rely on picometer shifts, where most of people rely on nanometer shift, but our band is so narrow that we can manage measuring this picometer level shift. Yes, yes,
right.
you are.
Right. And one more thing is you are able to you are going to use OSA, optical spectrum analyzer, on the other end.
Yeah, we can use an OSA, but there, of course, the resolution is limited. So finally, what you use is an FBG interrogator. So you probably know that there are a lot of companies that bring interrogators, and for instance, fiber sensing, micronoptics, and all these companies, and we rely on these specific devices. So FBG interrogators.
Great. Yeah. Thank you.
Thank you.
Thank you. Thank you for the good discussion. And is there any additional questions or comments?
I wanted to comment again that I think it's a very nice initiative that you have had. I thank you very much, the MDPI, and of course the organizers. It was extremely nice to see that in a time frame of 2 hours we can mix different presentations on a similar scope, which is plasmonic sensing, and the community there is growing, and it's a pleasure to see that from years to years. We see more and more people active in the field. We see that companies are emerging as well. And so it shows that the TRL is rising and it's reaching also commercialization stage. It's extremely nice. And I would be happy to see all of you for a real physical meeting in the future. I think there will be many occasions, starting from SPI Europe, BGPP, and biosensor conferences, of course. So we will have many opportunities to see each other in the future. But definitely this kind of meetings, virtual meetings, they are also extremely interesting, I think. So both are useful.
Yeah, I second the thoughts, Christophe. It's excellent platform to actually come together and share our thoughts and get to know each other more importantly.
Yeah. So that's extremely nice.
I fully agree with what Christophe said. It is indeed a very nice way of doing, and personally, I would like to thank Nunzio, Cennamo for this to put this together along with Charlotte and MDPI. So I think in overall this was a very nice initiative, and hopefully we'll have the next initiatives. And as also Christophe said, we can be in other places physically. That I already met some of you physically, so hopefully we can meet in somewhere. But this is also a good approach, because sometimes we cannot travel as much as we want, and we can. So having these online meetings in just 2 hours, as you said very well, 2 hours sometimes is the time I take to arrive to the airport and arrive to a place. So here it's a very interesting alternative to complement the physical initiative. So thank you everyone for your for this session. And I'm not sure if Nunzio want to say some last words or not. Now you have to say.
Thank you so much for this very nice community. I think this is a very interesting opportunity for next collaboration, for to develop a very interdisciplinary approach in the sensing, because I think in optical fiber last period is very important to use in an interdisciplinary approach to develop a new kind of sensor based on biomolecular sensing, based on new concept of transducer, very simple to use, very simple to realize, with high sensitivity, and the optical fiber can be used in this approach, I think. And this community, this context could improve the capability of this sensing approach. I hope I hope this is very interesting for the student, in particular for the PhD student, this context, to obtain a new generation of student that can be continued this approach, this school, this research school. Thank you so much for all. Thank you so much for all.
Thank you. Thank you to Rogerio for chairing this session and the speakers for their time and interesting contribution. Also, thank you to our attendees. I hope you enjoyed this live session. Just a quick reminder that this live session was recorded and will be made available on the conference's website as soon as possible. So if you want to check anything again, don't hesitate to go over the live session. Join us tomorrow for the second live session of this conference. And yeah, that's about it. Thanks, everyone. I wish everyone a nice day, morning or evening, wherever you are in the world. And take care, everyone.
Thank you. Bye.
Bye. Bye. Bye. Bye.
Bye-bye.